Good evening, and thank you for staying for the final talk. I'm sure all of you are kind of exhausted after the long day. I'll try to make this quick. My talk is about incorporating genetic heterogeneity into epidemic models for H1N1 influenza. And as we've seen in the previous talks of the day, one of the common methods to model diseases like H1N1 is the SIR model. I won't go into the details, but individuals go from being susceptible to being infected at a rate beta, and individuals recover at a rate gamma. An important assumption made by the SIR model is that of homogeneous susceptibility, infectivity, and recovery. But the truth of the matter is that heterogeneity exists. This is a map of the world, the number of laboratory confirmed cases during the H1N1 swine flu pandemic of 2009. This was a really severe pandemic. In the United States, between April 2009 and April 2010, there were over 12,000 deaths. And in this map, we can see that the number of lab-confirmed cases is very different in different countries of the world. And this could be because of many reasons. It could be that there is heterogeneity at the host level, that different people have different susceptibility to the pathogen. It could be heterogeneity at the pathogen level. The virus might have mutated as it went from one country to the other. It could also be heterogeneity in spatial mixing. Different countries might be more densely or less densely populated. In this work, we focus on genetic heterogeneity, both at the host and at the pathogen level. So the way we do this is we assume subgroups in the susceptible compartment. At the beginning of the simulation, there is one infected individual. The simulation ends when there are no more infected individuals left. Within a susceptibility subgroup, we assume equal susceptibility. Different susceptibility is assumed across different subgroups, but the recovery rate remains constant. So once an individual becomes infected, all the individuals behave in the same way. To model a system like this, we need to estimate the following parameters. We need to estimate the number of susceptibility subgroups, the size of each subgroup, that is how many people belong to each of these subgroups, and the beta value for each subgroup. So this is the beta value that I'm talking about. And the way we do this is to model the host pathogen immune interaction. This is how we incorporate genetic information. Now, when we think about a viral infection like H1N1, one of the most important arms of the immune response is the CD8 plus T cell or the cytotoxic T cell immune response. H1N1 is an intracellular pathogen. So when it's present inside a cell, it's impossible for a passing immune cell to recognize that a virus is present unless particles from the virus are presented to the outside world on molecules called class 1 MHC or HLA class 1. So what happens is a CD8 plus T cell binds to a cell expressing HLA class 1, rolls around the surface of that cell, exploring each of the presented peptides. And if any one of the presented peptides is recognized as non-self, an immune response gets mounted. In this video, we see a CD8 plus T cell releasing proteins called perforins and granzymes once it has recognized the presence of an infected cell. These proteins cause pores to be formed on the surface of the infected cell, which will cause its contents to bleb out and cause the cell to die. The molecules involved in this binding are the T cell receptor from the CD8 plus T cell side, the HLA class 1 from the side of the infected cell, and the peptide from the virus, which is also called epitope, shown here in blue. Here we see just the binding surface, just the binding site of the HLA class 1 and the peptide from the virus. This is the molecule that we will be focusing on in the rest of this talk. There are two conditions that must be satisfied for a CD8 plus T cell response to be mounted. First, the peptide must be presented by an HLA class 1 molecule, as shown here. And second, the presented peptide must be recognized as non-self. But it's important to recognize that this first condition has to be satisfied. If a person's HLA alleles are able to present only 1 or 2 or in fact 0 epitopes from the virus, it becomes impossible for the cytotoxic T cell to even recognize that a virus has entered the host. And therefore, their immune response will be very, very weak, if existent at all. On the other hand, if a person is able to present a large number of epitopes, it's extremely likely that the cytotoxic T cell will recognize one of those as a foreign epitope and mount a strong immune response. This leads us to this functional form, where the susceptibility of an individual K is inversely proportional to the number of epitopes that that individual presents. Now, an individual is not just a single allele. A person has 6 HLA class I alleles, 2 each of type A, B, and C. The frequencies of these alleles are available through databases like the Allele Frequency Net database, and we can generate HLA genotypes by sampling 2 A alleles, 2 B alleles, and 2 C alleles in a manner proportional to the frequencies that have been measured. When we do this, we can estimate the number of epitopes presented by an individual as a function of their entire HLA genotype. So we look at the epitopes presented by each of the alleles in that person's HLA genotype to give us the number of epitopes. But the number of epitopes is not something you can just read out. It's something you need to predict. And some of the methods out there to predict the number of epitopes are either learning-based methods, where experimentally determined peptide MHC complexes are used as training data, or energy-based methods, where the binding affinity between the HLA allele and the viral peptide are used to predict what the binding affinity will be between a new viral peptide and the individual's HLA alleles. In our work, we use 3 epitope prediction tools: the artificial neural network, stabilizing matrix, and combinatorial peptide library, and consider a viral peptide to be an epitope only if all 3 of these methods predict the binding affinity to be very, very strong. Now that we've been able to represent individuals by their HLA genotypes, predict the epitopes each of those individuals can present, we can get an estimate of the number of epitopes that each of the individuals in the population can present. We can now group together people who have similar susceptibility, giving us a very small number of susceptibility subgroups, even in populations with millions of people. We've found that the number of S subgroups can range from 1 to at most 10 to 20. It's not a very large number. Now, by doing all of these analyses, we have been able to estimate the number of susceptibility subgroups, M, the size of each subgroup, xi, which is the fraction of population in each of these subgroups, and the beta value for each subgroup. Actually, we have estimated the epitope value. Let's talk about how we go from the epitope value to the beta value. To do that, we recognize that beta in the SIR model is a composite of several properties: the infectiousness of an individual, alpha, the average number of contacts per individual per unit time, C, and SI, which is the individual's susceptibility. Note that only S is given the subscript I here. This is because we've assumed alpha and C to be constant across individuals in the population. Since SI is inversely proportional to the number of epitopes, we can write it as Z times 1 over AI. All of these 3 proportionality constants can be clubbed into the same constant Y. This Y can then be estimated using experimentally determined values for beta. Actually, this is a determined value for R0, as was discussed in the previous talk. We can estimate M, the number of subgroups, Xi, the proportion of people in each subgroup, and EI for this particular ethnicity viral strain pair. Plug all these values into this equation, leaving Y to be the only remaining unknown. Once Y has been estimated, the same Y is used for all simulations that we carry out. So the overall workflow is that we start with HLA class I alleles, represent an individual by their full HLA genotype and not just focus on individual alleles. We estimate the number of epitopes each person can present by considering the full proteome of the virus. This susceptibility is estimated for every individual in the population, allowing us to group individuals based on susceptibility, which allows us to model a slightly modified version of the SIR model. The output is the fraction of the population which got infected. Now, in our simulations, we represent a population using a vector called the susceptibility profile vector. In this vector, which we term SPV, every individual in the population is represented by their corresponding beta value. The inputs that we use are HLA frequencies for 61 ethnicities and H1N1 proteomes for 166 viral strains, of which 81 were isolated in 2009. The predicted value is the final epidemic size. Now, one of the interesting things we observe is that no single parameter of the susceptibility profile vector can predict what the final epidemic size will be in the presence of genetic heterogeneity. In this figure, we see beta, which is the average susceptibility of the whole population, and its relation with the epidemic size. The red curve here is the same relationship in a homogeneous population, if there were no genetic heterogeneities. We can see that when heterogeneity is present, the epidemic size can take values as low as 40% or as high as 100%, even for the same value of beta. We also looked at other parameters of the SPV, such as the standard deviation or the number of susceptibility subgroups, and we find that none of these individually can predict the final epidemic size. We find that, of course, some of these characteristics taken together correlate well with the final epidemic size, with the overall message that diversity in susceptibility protects the population against a larger spread. But we can go far beyond just epidemic size predictions, since we have worked with actual genetic information from both the host and the pathogen. Now, when the 2009 epidemic took place, one of the analyses that was carried out was the identification of high-risk alleles. This particular allele, HLA-A*24, was found to correlate with mortality rate due to the pandemic H1N1 virus. In our data, we find that this correlation does not hold true in general. In this table, we see the 3 ethnicities which have the highest frequencies of this particular high-risk allele. When we focus on these 2 ethnicities here, they have very similar frequencies of this high-risk allele. And yet, we see that their average epidemic size are very different from each other. This highlights the fact that it's important to walk past individual alleles and look at entire HLA genotypes, because an individual really does have 6 HLA alleles. Also, at the population level, we need to look at the full susceptibility profile vector to give us any accurate predictions of what the epidemic size will be. Another point that was analyzed was that indigenous ethnicities in general experienced more severe epidemics than their non-indigenous counterparts. And in our data set, we had only one such case, the indigenous ethnicity USA, Alaska, Yupik, and several non-indigenous ethnicities from the US. This particular relation held true for the ethnicity USA, Alaska, Yupik, irrespective of the viral strain being considered. So this shows us that host heterogeneity is an important factor. But once again, when we focus on 2 particular ethnicities from Australia, we see that the average epidemic size for the first ethnicity here is larger than the average epidemic size for the second ethnicity. But this trend is reversed when we focus on strains isolated in Australia. In this table, we see the first 2 rows corresponding to the ethnicity Australia Cape York Aborigine, which notice had higher epidemic sizes on average. But when we focus on viral strains isolated in Australia, the epidemic sizes are very low. On the other hand, this ethnicity has a lower epidemic size on average, but when we focus on viral strains isolated in Australia, the average epidemic sizes are much higher. So this drives home the fact that looking at only host heterogeneity does not give us sufficient information to make general claims about the epidemic size that a population might experience. This leads us to some of the main conclusions of our work. We find that both host and pathogen heterogeneity are important to predict epidemic size. We provide a method to do this by modeling the CD8 T cell immune response, which incorporates both host and pathogen data. We find that we are able to generate more accurate predictions of epidemic size. And the overall message is that greater diversity in susceptibility leads to smaller final epidemic sizes in general. We have ongoing work in which we also introduce spatial heterogeneity by taking into account a network of contacts. So in this work, we will be looking at genetic as well as spatial heterogeneity. This ongoing work will be presented tomorrow in the poster P5. Do come and take a look at that if you're interested in further discussions. The work that I presented here will also be presented in poster P6 with a greater emphasis on the correlations between SPV characteristics and final epidemic size. So please do come and take a look at that as well. The work presented today was published last year in PLOS Computational Biology. And this work was carried out in collaboration with Martin, Professor Nagasuma Chandra, who's my guide, and Carmen and Sumanto, and Gautam Menon from IMSC. So I'd like to thank all of them for their help and all of these agents for their support and funding. Thank you.